All right. Well, we can get started now. We're excited to have Jennifer Moore-Kucera here with us, who's currently the Climate Initiative Director with American Farmland Trust. She currently leads the Farmers Combat Climate Change program that focuses on regenerative ag practices, land stewardship, and renewable energy. And she's been a leader in soil health, soil biology research and outreach for quite some time, through positions with NRCS, Texas Tech University, Oregon State University. So we're really excited to get some soil biology foundations and some of the new science. So with that, you can take it away.
Great. Thank you, Joseph. And thanks to David for his great presentation. I really learned a lot from him. We're going to totally shift gears now and scale and look mostly below ground and discuss the role of soil organisms as key drivers of soil health functions important for crops and many of the above ground impacts that David talked about, as well as a number of other ecosystem services. So the goals I have for you today are to hopefully increase your awareness and appreciation for soil life and the influences on soil health. So when you walk across your field next, or even in your backyard, you can really appreciate all of the life that is below ground supporting all of the life above ground. I'm going to provide you a means to discuss soil organisms using 3 functional groups. And we'll be describing what we call 5 biological hotspots. We'll talk about what they are, how they relate to key soil ecosystem functions, and how to optimize their presence and persistence. And then we'll finish up highlighting some new soil microbial research that I gathered from either colleagues that I've worked with over the years or new research that I have found keeping up with the literature as it relates to soil health, function, and management decisions. So I always like to start with some slide that emphasizes how diverse and vast the numbers of microbes and other organisms that exist in the soil. And this slide kind of does that. It has a lot on it. On the y-axis here is the number of living organisms in a cubic meter, which translates to a little over a cubic yard of topsoil in temperate regions. And then we have different types of organisms on the x-axis here. So in a 1.3 cubic yards, there are over 10 trillion bacteria, 100 billion fungi, and sometimes they're not counted, but rather how many miles or feet of their fungal networks exist. And in just a single teaspoon, a healthy teaspoon, we can have hundreds of feet of actual fungal networks, which is a little staggering to imagine, but true. There's up to 100 million algae, 1 million nematodes, 100,000 springtails and mites. And you can see as the organisms get larger, their numbers get smaller. In addition to just the vast quantity of soil organisms, they also carry mass. And so in about a hectare, which is about 2.5 acres, if we add up all of these organisms, it's equivalent to the weight of about 20 cows. And that's a pretty conservative estimate. I've seen other estimates where a single acre carries in a healthy soil can carry the equivalent weight of below ground biomass of about 20 cows. And so this really translates to soils hosting over 1/4 of the Earth's biodiversity. The majority of those organisms, especially the microbes, have not been cultured or fully identified. So we don't even have a great handle on who's there, what they're doing, but advances in molecular tools especially are really increasing our understanding of these organisms and the plant microbe and animal interactions that are involved in all soil processes and functions. So why should we manage for soil biology? This is kind of a little bit of a brief overview, but when we choose management practices that increase the amount, types, and availability of plant residues and minimize soil disturbances, one of the results is an increase in both the numbers and types of organisms that are supported. Each of these organisms perform critical functions that maintain and support the health of the ecosystem above it. For example, as microbes and other soil organisms process those organic materials from plant residues and animals, they partially transform them to carbon dioxide, which is released to the atmosphere, but some of that carbon is converted into stable forms into the organic matter and sequestered in soil. So in doing so, soil microbes play a really key role in mitigating climate change and overall ecosystem resilience. Soil microbes are also involved in helping form and stabilize soil aggregates that improve soil structure and resist erosion, thus protecting air and water resources. Enhanced structure and increased organic matter also increases the amount of water that can infiltrate or get into the soil, reducing flooding potential, and also water storage capabilities, which is important in the summer months as things start to heat up and dry out to provide water for plants. Additionally, during the decomposition of those plant residues, plant available nutrients are released by soil microbes through a process called mineralization and also through predation. And it's the interactive and synergistic effects of all of these functions that help to support plant growth, to provide food, fiber, fuel, and additional ecosystems for all life on Earth. So that's just a snapshot and an overarching view of why soil biology is important, especially for crop production, but also for critical ecosystem services we depend upon. So there are a huge number of ways that we can categorize or classify soil organisms. And again, really emphasizing that the soil biological community is controlling all of these soil processes and functions important for plant productivity. Some ways scientists classify them are by size, molecular approaches, their trophic level, or what they eat, what their energy source is, and other means. But keeping in mind that soil biodiversity, and functions are complex interactions between inherent soil properties, climate, management, and the life within the soil, a real simplified view is to categorize soil organisms into these three broad functional groups. They include the soil ecosystem engineers, the biological regulators, and the chemical engineers, or the microbial decomposers and transformers. The following slides will talk about what these groups are primarily responsible for. I'll give you some key representatives that will be identified in a brief overview of some of the functions that they're involved in specifically and where they're found in soils. Some of those key ecosystem functions are highlighted here: the decomposition and carbon cycling, that building of and creating soil aggregates and pores, regulation of the population through biological means, that nutrient cycling I already talked about, and plant productivity. And all of these work together to improve the resiliency of the ecosystem, to help it deal with threats from climate change and other stressors throughout the years. And so the arrows, you can see it's complex. There's not any one type of organism, isn't specific for really any one particular function, but they work collectively as a community. And when you have a diverse and active community is when you get the best functionality. So let's start with the ecosystem engineers. These are the larger organisms, sometimes called the macrofauna, and they include earthworms, ants, termites, centipedes, beetles, etc., as well as plant roots. These organisms help to move and redistribute and mix organic materials, and in doing so, they modify and actually can create their own habitat. So they're the movers and the shakers. They're taking materials, they're mixing, shredding, increasing surface area, and stimulating a lot of important processes. One outcome is the formation of really large pores in soils called biopores. These are really important to channel air and water that helps with drainage and aeration following a rain or irrigation event. These organisms, especially earthworms and plant roots, are involved in the building of those resistant soil aggregates. And by regulating resources and redistributing and access to these resources, these larger organisms create hotspots that support high numbers of the smaller microorganisms. And they also help transport those organisms as they take residues from the surface, bring it below ground, mix it. They're also transporting and serving as microbial highways. And just like a fun fact, earthworms have been accounted for turning over the top 6 inches of topsoil within 10 to 20 years. So they can really transform and help to build organic matter and break down those residues for other important functions. The second group are the chemical processors, the chemical engineers. I've even been known to talk about them as the chefs in the military of the soil. These are the microorganisms in the soil. They're the smallest and most numerous. They include organisms with the bacteria, archaea, fungi, and single-celled organisms called protozoa. They don't look like much. They just look like little round rods or circular spheres. The fungi look like little threads through the soil, but they are responsible for regulating over 90% of the energy flow in soil. They are the transformers. That's why I call them the chemical engineers. They take carbon from the plants and the animal residues, and they create amazing byproducts with it, including organic matter, important for carbon sequestration. They also help to release plant nutrients and plant stimulating biochemicals, hormones or other plant growth promoting substances. The last group are the biological regulators. These include small invertebrates such as those protozoa again, also nematodes, potworms, springtails, and mites. And through grazing, predation, or parasitism, these organisms help to regulate the population of other invertebrates and microbes. About 1 hectare, or again, those 2.5 acres of soil, contains the equivalent weight of 2 sheep of protozoa. So if we added up all of the protozoa, this is an image of one over here on the left, they would have the equivalent weight of 2 sheep. So they're very, very tiny, but when we add them all up, they have a significant impact on biomass. These guys love bacteria, for example, and the green blobs inside of this organism have been dyed to show the bacteria that this organism is consuming. They've been known to eat hundreds of bacteria every minute and millions or billions in a day. Most soil nematodes always get the bad rap. They're cause of major crop problems and parasitism and pathogens, but the majority are not the bad guys. They are mostly good guys in soils, and the predation is responsible for up to 20% of soluble nitrogen in soil. They also help to regulate other organisms, as I'll show in a minute. So it's really important to create a healthy, diverse community that can naturally keep the checks and balances of organisms in place. These biological regulators are fiercely competitive and again are keeping those populations under control. They also help with plant growth. So this is an image showing the protection from a pathogen called Rhizoctonia solani. And in this left-hand side, the roots were grown in the presence of organisms called springtails. And on the right-hand side, they were grown without springtails. So you can see this nice healthy root on the left-hand side. It's somehow interacting with the microbes in soils, and it's helping to protect this plant from pathogens. One of my favorite organisms are nematode-trapping fungi. So this is a nematode blown up in a scanning electron micrograph, and this filament with this noose around it is the fungus. It basically creates a noose and it attracts this soil nematode as it's swimming through soil, and basically the nematode swims through that noose. It creates some kind of biochemical signal to the fungus, and it constricts down on that nematode, trapping it, and then begins to suck the life out of it. Another example of a nematode being attacked is on the right-hand side with a mite just preying on one of those nematodes. In the lower left, I just really like the name of these guys, the Vempriliads. They are protozoa. And these guys are shown eating a fungal root pathogen that's involved in take-all disease. So they come in, they puncture the fungus, the fungal hyphae, and basically, again, suck the life out of that guy. I already saw our friend the protozoa eating billions of bacteria a day. And then this is an example of a soybean cyst nematode that's being parasitized by a fungus. And so there's little tiny thread-like substances, and it's releasing chemicals that's parasitizing this nematode, or yes, this nematode involved with plant pathogen. So it's really competitive. It's something that if you believe in reincarnation, you do not want to come back as a soil microorganism, because it is a very fierce competitive world below ground. Everybody's trying to eat everybody else. And you know, I kind of joke, I'm a microbial ecologist by training, and I tend to look at the world microbe-centric. So I really like this figure, this video that I found online where they did a 15-week time lapse. And on the left-hand side, it's without the presence of soil macrofauna. So those earthworms, the beetles, the centipedes, springtails, mites, etc. Just fungi and bacteria are allowed to grow on the left-hand side. On the right-hand side, they were allowed to persist. And you can see how quickly the materials are being decomposed on the right-hand side. You can actually see them mixing and bringing materials from above ground, below ground, creating channels, incorporating organic matter, transforming these carbon-based and substances on top and releasing plant nutrients during the process. So it really helps me remember that it truly is a community effect, and we have much better functions and cycling of nutrients and formation of those aggregates when we support those larger organisms on top. I know we're talking about crop production, and this is in a forested ecosystem, but the same thing happens when we build that plant residue on top through cover cropping and reduced disturbances. That crop layer is also being attacked by these macroorganisms and distributing it. So very, very important to have that layer and to support those organisms, because once they break down, as you could see in the previous video clips, the decomposition slows down dramatically. So are numerous factors that influence biodiversity and the activity of soil organisms. And although microbes can exist in the most extreme environments possible, most agricultural soils benefit from a much more narrow window of conditions. And I often kind of talk about it as the Goldilocks syndrome, right? Microbes don't like it too hot or too cold. They don't like it too wet or too dry. They don't like it, you know, too acidic or too alkaline. They like conditions just right. So for agricultural systems in general, there are differences depending where you are and what crops you're growing. But overarching, they like near neutral pHs around 6 to 7.5, warm soil temperatures in that 60 to 90 degrees Fahrenheit level. Again, this tracks very well with the optimal temperatures and pH levels of the crops grown, as David discussed in his talk as well. For moisture conditions, they like field capacity, so that amount of water that's left over after about 2 or 3 days following a heavy rain event, for example. And that's because then there's good aeration as well, the balance between how much water is left and how much air in the soil. Of course, they can't do anything if they don't have food and energy sources, so they like abundant and diverse food sources, and also diverse soil pore sizes. So again, it's the Goldilocks. They like it just right, and conditions have to be ideal. And this translates to the majority of life in soil not being in an active state, but they're rather in a resting state that allows them to stay alive when conditions are not just right. However, the good news is that they rebound quickly when conditions improve. So for example, over 90% of bacteria in soil are often in an inactive state because they have not been able to move towards an organic substrate to use, but spring to life quickly upon re-wetting of soil to redistribute those soluble organic carbon and nutrients. So if we want to encourage a diverse and healthy habitat for microbes, we have to provide the food, nutrients, proper aeration, pH, water, etc., for them to thrive. So how do we do that? How can we manipulate the soil microbes and other organisms for our benefit, for crop production and ecosystem services? There are different plant species, varieties, and plant growth stage that select for different microbial populations, and scientists are just beginning to understand how plant species and varieties affect those. There's really cool cutting edge work on fertilization impacts and those impacts on root exudation chemistry that also influence the composition and quantity of the microbes that are present and how many there are. A lot of really cool and interesting work on soil amendments, including biologicals. These have been known to stimulate those beneficial microbes to defend against plant diseases and pathogens. Some of them have been associated with fighting off drought, for example. Really, really cool work. However, because of that competition, because of the complex nature of soil particles and its chemistry, they're really fraught with a lot of issues with consistency in delivery and maintaining those biological amendments. So lots of cool work, but they need more research. We can also manage the environment. That's through some of our management practices for soil health to minimize stress through reducing pathogens, drought, temperature extremes, etc. So how do we manage the temperature environment, that moisture environment? And really it comes down to maximizing the presence and duration of these biological hotspots that I'm about to talk about. But before I do, I just wanted to note that if you add materials that suppress certain organisms, or if you actually add certain organisms to a soil, it could potentially lead to an imbalance in the system. So when you're trying things out, don't do things more as an insurance practice, but rather with specific purpose. So by knocking out one threat, you might actually be creating a new threat. So it's important to work with local experts, people who have used these products on farms, and have controls when you're trying out new products. I find farmers to be the best scientists available, willing to try a lot of new techniques and approaches. So we just want to learn from each other as best we can to find out what works the best in our specific systems and environments. Okay, so these biological hotspots. So despite the fact that most microbes are in a resting phase, these hotspots are where population size and activity can be orders of magnitudes higher relative to what's found in the bulk soil nearby. So a highly functioning soil will support all of these hotspots that I'm going to talk about, sometimes called spheres of influence. These hotspots include the area immediately surrounding plant roots, also known as the rhizosphere, where soil microbes are stimulated by the release of sugars from plant roots as an output of photosynthesis. As plants produce above ground biomass and die, their residues will form a litter layer. As these residues are decomposed, earthworm activity and root channels support increased biological activity and aid in the formation of those macropores or biopores and strong, stable aggregates. These hotspots support multiple biological processes that are working all in concert together. So if we can select management systems to optimize and maximize these hot zones in soils, soil biological function is enhanced, and that results in more efficient and resilient systems. So the following slides will run through each of these independently. They're also just kind of FYI, they have other fancier names that are associated with them. I think rhizosphere is a common term used in the industry, but some of these others are a little bit less common. So I like to just talk about them from what their hotspot is, the pore spaces, the litter layer, these earthworm and root channels, and aggregate surfaces, because I can't even pronounce this word, to be honest with you. So let's go through these one by one. So a hot spot for the ecosystem engineers, those larger organisms, the mixers, the movers, the shredders, is that litter layer again, like in that video of the forest. Here it is in cropland. So this zone, the litter layer, includes recognizable plant and animal residues that are undergoing different stages of decay. There are higher concentrations of those larger organisms, and also the smaller organisms that help further break down the residues after these larger soil animals shred, mix, fragment the residues. They increase the surface area, allowing for the microbes to come in and transform them into other products. It also, from a above ground perspective, it protects the soil. So this is our armor. This is what protects the soil from the impact of raindrop or irrigation water. It also helps to moderate soil temperatures from wild fluctuations. It conserves soil moisture, and of course, because of its rich sources of carbon and nutrients, it's energy sources for many organisms and soils. So it's very critical, both from physical perspective as well as a biological perspective in soils. The next hot spot is for the ecosystem engineers. Again, these are typically plant roots in earthworm channels. Again, they're mixing and moving residues, and they form these really large pores that help with drainage and aeration. Also, the earthworm pores in particular, as shown here on the right-hand side, are incredibly nutrient rich. They're microbially enriched with many, many orders of magnitude of bacteria and fungi present here. And then it actually forms a environment that is very conducive to plant growth. So this is a soil image. The dark channel here is left over from an earthworm channel. You can see it's dark. It has a lot of organic matter. It's very stable, lots of good pore space in there, and lots of microbes. That creates a perfect environment for this plant root. It's now at an advantage to a root that's grown nearby without this biopore and microbial inoculant, if you will. It's going to have an advantage to get a jump start from a nutrient perspective as well as water. So the sustainability or the persistence and how all these biopores are connected are very important, and all of that is disrupted when we use intensive tillage production, where we're really mixing and turning over the soil. All of those connective pores are now fragmented, and so water can't drain as well. The roots can't find those paths as easily as they can when systems are less disturbed. So another really important component in soils, a hotspot, is just the space between the solid materials, the organic matter, as well as the minerals in soil. So a healthy soil tends to have close to half of its volume occupied just by space. And this space is really critical. It serves as the lungs and circulatory system of the soil. In this zone, air and water flow are regulated, nutrients are cycled and transported in soil water. It's a site of microbial entanglement and sequestration of water and nutrients. And it's this mix of different sizes and arrangement of pores that also serve as biological highways, as many soil microbes can only move with soil water and water films. Other soil organisms can hitch rides on the more mobile organisms, or fungal hyphae, which tend to be prevalent in pore space. So in contrast to this healthy soil, where half of the volume is occupied by space, and then ideally half of that space is occupied by water and the other half is occupied by air, when we compact a soil, we actually squeeze out the availability of that pore space. And that's illustrated here. So now in a volume, we have much less space for water and air to flow through the soil, to be stored in the soil. And so that increases our flooding potential. It increases our anaerobic sites. and that can cause additional problems and propensity for pathogens. So really, really important to support the space between the particles. So the space between the particles are formed from soil aggregates. And aggregates are an arrangement, a three-dimensional arrangement of both the minerals, the sand, silt, and clay particles, as well as organic matter. And so these aggregate surfaces are also a biological hotspot. Again, they help create stability and resist erosion. Wanted to highlight again that they're both the minerals as well as organic materials. These aggregates also, depending on the shape and the configuration, can help protect organic matter. So pieces of organic matter can get trapped inside of those, kind of preventing the microbes from accessing it. And it's a form, an important form of carbon sequestration in soils. And the aggregate itself is a microbial habitat. Of course, without the aggregates, you can't support the pore spaces, so very important. And although they're often taught in soil science classes as a physical process, it's actually a really important biological process that's created in part by microbial glues, fungal hyphae, roots, and dead cells. And so when undisturbed, these soil organisms transform residues and create organic matter. And so there's two processes that form soil aggregates. One is through physical enmeshment, and that's primarily through fungal hyphae, which you can't see here, but you can see the roots that are acting like a net, or I think of it as a hair net, right? When you're working in the food industry and you keep all of your hair in place, the fungal hyphae and the roots act like a net that encapsulate and enmesh soil particles, keeping them in place and protecting them. There's also, so this is just another example. This one does show the fungal hyphae, these white threads through the soil, and you can actually see that they're connecting particles across this little clump of soil that I have. I like to think of it as, the Spider-Man, the superheroes of the soil that are helping hold those particles together and keeping that bus from falling off the edge of the bridge in Spider-Man. Sorry, I've got kids. And then also there's a biochemical process that is actually happening. So this is a scanning electron micrograph. It's actually bacteria, and these are soil particles. And you can actually see these gooey substances that the bacteria release that help to stick them to the soil particles. Fungi also release glycoproteins, for example, that are important in helping to stick the soil together to resist erosion. And again, this is another filament. These are bacteria, but fungi also do it. Two soil particles being bridged together by the filaments formed by special bacteria called actinobacteria. So again, tillage tends to reduce the overall number of soil organisms, especially fungi that form those hyphae, and the earthworms, important for aggregation and other soil functions. It also tends to favor increased bacterial populations and their predators. In contrast, no-till or reduced till favors higher overall populations, especially those fungi and earthworms, those larger organisms, important for creating and mixing those materials through that video I shared with you. So what does this translate to in the field? What this translates to is if you have poor aggregation in compacted soils, that happens more commonly in conventionally tilled soils on the right-hand side compared to a system with no-till on the left-hand side. The water can't infiltrate. We collapse that pore space, we reduce the sticky substances, we're reducing our microbial population, important for that, and the water can't get in. So this on the right-hand side is the slump test that you can do with a sink strainer. We can talk about that later. But this one on the left-hand side is good aggregation. It's holding together. It's holding on to that water. On the right-hand side with poor aggregated soils, it just slumps, it falls down, it doesn't hold up, and you can actually see the water is going to be moving away and all those particles along with it. So the last hotspot, and these are important for those chemical processors, the microbial decomposers and transformers, is the rhizosphere. And that's because some of the carbon that plants are taking out of the atmosphere from CO2 are being released below ground in sugars and other substances that help stimulate the microbial population. There's a lot of symbiosis that's happening. It can translate into the protection of plants. There's a lot of chemical signaling and communication that's going on, nutrient cycling, and again, that overall resilience. When you have healthy plants, healthy roots, you stimulate those microbes that are going to help create all of these benefits for you. So just again, a summary of the organisms found there. The bacteria, they typically are the most numerous. And collectively, all the microbes make up anywhere from 2 to 5% of soil organic matter. So it doesn't seem like a lot, but again, they're responsible for the majority, 90% or more of all the energy and carbon flow in soils. On a mass basis, there's about anywhere from 1/2 to 3 tons of bacteria per acre. In contrast, the fungi tend to outweigh the bacteria. They're a little bit larger, so that makes sense. And in a healthy soil, their biomass can go up to about 5 tons per acre. Similar to bacteria, there's both saprophytic, those are the organisms breaking down dead stuff, and also there are pathogenic organisms. Unique to fungi are the mycorrhizal organisms that we'll talk about in a minute. Those are symbiotic and help plants immensely. And then also wherever there's these microbes, they're the regulators. So protozoa and nematodes are there to consume the microbes, recycle nutrients, and help keep populations in check. And then just a little fun fact on the right-hand side, for every 1% organic matter, that translates to 10,000 pounds of carbon, about 1,000 pounds of nitrogen, of which about 50 pounds of that is held in the microbial biomass nitrogen pool. There's 100 pounds of phosphorus that will be released through microbial action, and that again, 5 pounds of that is embodied within the microbial biomass. And when these organic residues are decomposed and mineralized, they then release those plant available forms such as ammonium, sulfate, and phosphate. My dog is really excited about this, if you can hear him in the background. One of the symbiotic plant microbe relationships I just wanted to highlight are legumes and nitrogen fixing bacteria. So the nitrogen cycle is propelled in part by these specialized bacteria. It's one of the most important systems in the maintenance of terrestrial life. Living organisms produce the vital organic compounds, those building blocks of life, and the amino and nucleic acids that require nitrogen. So how this works is the plants then support these bacteria that infect, but in a good way. The plant roots, they form a specialized nodule that helps to protect the bacteria. And inside the nodule, these microbes, these bacteria, take nitrogen right out of the atmosphere, right? Our atmosphere is primarily dinitrogen gas, inert, doesn't do anything for anybody. But these bacteria can take that dinitrogen gas, split those really strong triple bonds between those two molecules, and convert it into ammonium. And then that becomes a source of nitrogen for the plant. So really important. The plant shuttles sugars to the bacteria, and again, that protection, and in return, the bacteria supplies the plant with nitrogen. Probably the most important ones in agronomic systems are these symbiotic relationships that involve the dependency upon the bacteria with the legume. But there are also free livers that live in soils, so they kind of get close to the roots and take advantage of some of that carbon that's being released, and also fix nitrogen. Okay, so that's one of the most important symbiotic plant microbe relationships. A second one is the relationship between plant roots and now fungi, called mycorrhizae. So it's a Greek word, mycos meaning fungi, rhiza meaning roots. And in this case, plants use anywhere from 5 to 20% of the carbon from photosynthesis to, again, feed the microbes, in this case, the fungi. The fungi then act like, now they're not acting like a hairnet, but they're now acting like hair extensions or root extensions that can increase the absorptive surface of at least 10%, of seed estimates up to 40% or more. And that absorptive surface now has access to more water, more nutrients that can then be brought back through the fungal hyphae to the plant for uptake. They're especially important in phosphorus acquisition and zinc. Also, these fungi are known to release chemicals that suppress plants and diseases, so help to protecting plants. And again, they help build those networks and that net-like structure to help form stable soil aggregates. So two really important symbiotic associations. One is plant roots and bacteria with nitrogen-fixing bacteria and legumes, and then the mycorrhizae fungi between the plant roots and the fungus. And then I want to now shift from the hotspots, what they are, who's there, why it's important, to some examples from the literature of how microbes and the management helps plants deal with stress and also to support plant growth. This is just a really nice slide that I found from a PhD student at the University of Queensland in Australia. And it just summarizes the importance of microbes that have been identified to help plants deal with temperature, drought, waterlogging, insects, pests and pathogens, soil toxicity, and nutrient limitations. A couple of examples are shown here where this student added soil microbes to corn plants grown in a 2-week period. And on the left-hand side, there's no microbes added. And on the right-hand side, there's microbes that have been added. And you can see the plant growth differences. This is with a special bacteria that's known to promote plant growth. Of course, this is done in the greenhouse, and all those competition factors kind of weigh in and create some inconsistencies with results in the field, but it just emphasizes how important microbes are to plant growth. Another really cool study, I actually found this last night while I was preparing for this, is looking at crop management history and how that influences the soil microbes, plant growth, and system function. So in this case, they compared soils that had been historically monocrop to just a single crop year after year, and compared that to soil in rotation. And so I don't know the details of what those rotations were, but then they grew peanuts in these two different soils. And so this is what it looked like with the rotation soil, and this is with the monoculture soil. And they found that the plant weight and also the length was much higher in the peanuts that were grown in the soils that had that rotation history compared with the monoculture rotation. They also found that the diversity of the bacteria was lower in the monocropped versus the rotation soil. And then they looked at the genes that are responsible for nutrient cycle, for plant protection, et cetera. And they found that the genes were underrepresented. There were fewer representations, the best way I can describe it, involved in the plant growth promotion in the monocrop soil compared to the rotation soil. So how we handle our soils has long-term impacts. So this is just taking soil from 2 historical management systems and growing peanuts and seeing how they respond. They also found that the history was really important when they then took the peanuts and grew them in sterile media. And then they inoculated the plants where they were exposed to the bacterial suspensions from both the monocropped and the rotation. So this is just extracting the bacteria from those 2 systems and then growing the peanuts. And they again found that the peanut growth was reduced in the plants that are exposed to bacterial suspension from the monocrop versus the rotation. So really, really neat work that indicates the importance of management history. Another really cool is the communication. This is, I love this slide. It's showing how a tomato plant or a plant is being grown. And in this case, above ground, they're being attacked by aphids. And then the plant is sending out a a chemical signal into the air that attracts these plant parasitic wasps that come in like the Air Force to rescue the plant, and it helps to consume and prey on those aphids. Another below ground relationship, in this case, the plant is being attacked by a pathogen from above ground through the stomata. And in this case, the plant sends a chemical signal below ground, and that translates into the swarming of bacteria, again shown in this fluorescent green surrounding that dark root. The bacteria come in, they swarm the roots, they help protect that plant because it's being stressed from above ground so that it can't be attacked below ground by any other predators or pathogens. And then those bacteria also send a chemical signal back up through the plant that tells the plant, close your stomata, the bad guys are in town, right? Protect yourself. And so there's a communication between the plants and the microbes and how they interact when a pathogen is present. And then there's another example where, again, the plant on the left is being attacked by the pathogen. And then this plant on the right is a healthy plant, not being attacked. But there's a communication network through the mycorrhizal hyphae, those filaments that can connect the fungus to the plant roots. And so this plant is being attacked. It sends a chemical signal through the mycorrhizal hyphae to the nearby plant, telling them, bad guys are nearby, right? Close your stomata, shut down, lock the doors, right? Lock the windows, and protect yourself. So this communication is amazing. And scientists are just beginning to decipher it and decode it. The impacts, you have to have these organisms nearby, you have to have the stimulation of the bacteria, the predatory wasps, etc. And if you have that, you can increase natural pest control, increase your disease susceptibility, and decrease the use of pesticides that are being used. Okay. So then another example is just different crops select for different microbes. So in this study from ERA scientists in Fort Collins, Colorado, they grew potatoes in different cover crops and artificially inoculated them with a pathogen, again, Rhizoctonia solani. And they saw different disease development when potato was grown following these different cover crops. And basically they found the lowest disease severity was with the sordan, the sorghum Sudan grass cover crop species. And so they started to then look at the microbial community to identify who was there and what compounds they were being released to help provide this protection from the disease. And basically they found a particular gene that's involved in starving that Rhizoctonia from iron. And so when this particular organisms are present and this gene is present, the disease index and severity goes down substantially. So now they're translating this into the field and they're looking at how does this sorghum Sudan grass translate into disease protection in potato, and really encouraging results from that study, hopefully being published soon. Another example is with different cover crop species selecting for different types of fungi. So here's clover, oats, vetch, and wheat, and the different colored bars are different functional groups of fungi. So arbuscular mycorrhizal fungi, those symbiotic mutualists that team up with plant roots, tend to be highest in clover and vetch, whereas overall diversity was higher in the wheat system. And saprophytic organisms, those organisms that break down dead residue, or, dead residues, were higher in the oat. So by choosing different cover crop species, by choosing different varieties, we influence what microbes are there. And so scientists are looking at designing rotations as well as cover crop mixes to stimulate certain microbial communities for impact. Kind of not well studied, but this is one study where they looked at increasing rates of nitrogen fertilization and its impact on root exudation. So when roots are growing, they release different chemicals, and you can see again, different bars are different kinds of chemicals and different quantities of each. So as nitrogen rate increased, the composition of the exudates changed. And then this basically translated into impacts on that microbial community, differences in microbial biomass that translated into differences in nitrogen use efficiency for the plants, pathogen resistance, and overall plant growth promotion. So lots of really interesting ways to look at nutrients from a different perspective from the interactions with microbes and how that translates to overall plant productivity. Okay, I gotta let my dog out before he completely freaks out. 1 second. Okay. Sorry, he's driving me crazy. Okay, this is a study out of Washington, Mark Mazzola's work on replant disease. It's a little bit dated, but I really like this study. This is the height of an apple tree grown in replant soil. So the microbial population gets acclimated after long-term, growth of apple trees. And so when you try to replant that orchard to new varieties, you can often run into problems. This is the height of an apple tree that was grown if it was in virgin soil, just to give the perspective. What they looked at was mustard seed meal, and they found that it altered the types and numbers of the fungal community, and it provided different levels of control. So the mustard seed meal is this BGSA species, and this is the number, in this case, of the organism that's associated with replant disease, and how significant the numbers declined when you added the mustard seed meal. And this is the typical chemical that's added, telone. And you could see what happens in the first year. Both work pretty well. They suppress that organism and suppress the replant disease. But then in the following year, look at this rebound from after the chemical use. It's actually worse than it was before, whereas the mustard seed meal is no different from the control. And then finally, that is persisted 2 years later, where the mustard seed meal continues to have efficacy against that pathogen. And then that also translated into increased yield for this particular orchard. I'm going to skip that slide. This was also a study I found yesterday, and it shows again the importance of macrofauna, in this case earthworms, that act as biochemical reactors to convert labeled plant compounds into stabilized soil microbial necromass. What a mouthful. But necromass is basically the dead cells of bacteria and fungi that get absorbed to particle surfaces. And what this really translates to is all of these aggregates, these mineral particles, the circles of particulate organic matter, these extracellular, these are the gooey substances that are released that help stick particles together. And also microbial cell walls are really enhanced and increased when the system supported the earthworms compared to the system without the earthworms. And what that translated to was an increase in carbon sequestration and all those other amazing functions that we talked about earlier as well. And I just point that out because really the traditional views of soil organic matter always focused on the that it originated from the preservation of complex plant compounds, right, that are resistant to decomposition. But new research really emphasizes the importance of that microbial necromass, dead cells, and in the stabilization of organic matter that persists in soils for hundreds, if not, thousands of years. So how we manage our microbes is critical to carbon sequestration and building carbon in soils. That was just a little side note there. Couple more slides that just again emphasize what people are looking at and the influence and impact of microbes. This one is on increasing the perennial crops in a rotation, so converting from an annual crop to, in this case, a 5-year pasture. And what they found was that it increased microbial biomass and also soil carbon, up to 20% increase in soil carbon in a 5-year period. And what they found was that it was driven by labile carbon. So easily degradable sources of carbon are really critical to stimulate that microbial population and to help form that stable carbon ultimately. So you need both. You need the label to keep things going. It's sort of the fuel in the wood stove, if you will, to stimulate those microbes, increase their biomass. And then in turn, they will, through increased efficiencies, create those stable forms of carbon important for sequestration, as well as water storage and infiltration. And then to remind us again that it's a community effect, and both climate change and intensive land use impacts also how these organisms are growing. This study indicated that climate change and intensive land use reduced the actual body size of some of the soil fauna, the soil organisms. Intensive land use decreased their density. And what the researchers were suggesting was that this could threaten ecosystem functions like organic matter turnover, like nutrient cycling, like that population regulation that I mentioned earlier. And so we not only have to think about how our management is impacting these organisms, but also climate change and how we have to protect our systems as best we can to prevent these negative impacts from occurring. So what do they need? It's pretty simple. They need some food and they need protection. So how can we feed that below ground life? You choose practices that provide diverse, near continuous inputs and build reserves or organic matter. How can we provide and protect habitat? Choose practices that minimize disturbance of the habitat, just the aggregates, and the food sources, so organic matter and plant and animal residues. Choose practices that support a stable habitat from major swings in temperature, water, and chemistry. Again, that litter layer is so critical for doing that. So that's all wrapped up into really 4 relatively simple principles proposed by NRCS. And when we were there, we put a lot of these slides together. It's from our actual soil biology training module that I shared with you today. And this basically summarizes those 4 principles: minimize disturbance, maximize cover, maximize diversity, and maximize continuous living roots. Some people prefer to use optimize instead of maximize, and I think that that makes sense in a lot of conditions. But really it boils down to 2 simple rules, right? Protect and feed. You want to protect the soil through practices like no-till and reduced till, controlled traffic, avoiding tillage when wet, of course, implementing a sound IPM practices. You want to maximize that cover, the armor of your soil through mulching, again, conservation tillage, forage and biomass planting, cover crops, etc. And then you had to feed them, and feeding them with just the same type of carbon source year after year really depresses the microbial community and their functionality. So mixing it up through cover crop mixes, through crop rotations, through incorporating perennial vegetation into the rotation, incorporating livestock when appropriate into the rotation can really help stimulate that microbial community and other life underground that can help fuel them. Okay, and then just kind of wrapping up here, most agricultural soils are still losing rather than gaining carbon. It really comes down to managing for carbon. Again, we need both labile sources, those easily decomposable components that stimulates microbes. That primarily comes through plant roots and the exudates as they are growing. Carbon is the fuel for underground life. The soil life increases efficiencies of your operation. It increases the stability and resistance and resilience to disturbances. And really it comes down to our food security, climate resiliency. Our local and national economies really depend upon how we manage the soil microbe system. And I just throw this slide up to say that there are always windows of opportunities at the landscape. So how we manage it in exact field is important, but how we manage our field borders through increasing, restoring wetlands, riparian forest buffers, filter strip, grass waterways, windbreaks, etc., can really help that overall functioning at the landscape level. So the biological hotspots of life, action, and function are your rhizosphere that stimulates those chemical microbial processors. The litter layer is so important for protection and supporting our ecosystem engineers. The aggregate surfaces and the space between is so important as well, stimulating microbes and other organisms, and supporting and maintaining the earthworm and root channels, so we have these big channels for air and water. Again, that lungs and circulatory system of our soil. And all of those then help to support biological regulators, keeping populations in check, reducing inputs, reducing sources, and increasing the bottom line. So I think I'm going to skip that last slide because I just said everything that I've been talking about. Thank you for your attention. It was really fun to talk about soil biology again, and just encourage you to get your heads underground, listen to the soil, and yeah, soil biology rules. So thank you very much.
Awesome. Thank you so much, Jen. Give you a virtual round of applause, and I'm sure everyone else is appreciative too. I really like the Spider-Man analogy. That was awesome. And I'd never heard Aggregates.
I can't say it either. It's,
yeah.
Aggregatosphere, I think is how.
That's fun. I guess just because it's fun to take a question, if anyone has a question they'd like to ask, we have time for one question before moving on. But again, want to thank Jen for taking the time to share this really, really spectacular presentation with all of us.
Thank you. And I know I went up all right up into the end there. My email's listed. If anybody has any questions, I'm happy to discuss anything. And then some with anybody, reach out to me anytime. Thank you.
Well, it looks like let's try this one. Maybe you have some insight to share. So it was a kind of a specific question, but can Rhizobium bacteria last for 2 years in the ground if we are planting hairy vetch cover crop in the field every other year? So. Yeah,
well, I hate this answer, but right, it depends is the short answer. It depends on the soil, how long you've been doing it. Typically, it's recommended to inoculate multiple years in a row until you can build those populations and sustain them. That changes dramatically if you have low pH soils, for example, or other stressors in the environment. So I would, recommend reaching out to local Cornell Extension or local NRCS staff to really know the best way forward. But in general, in most circumstances, once those populations are built up, you can reduce the inoculation. But I wouldn't do so just haphazardly. Work with somebody locally.